The Cambrian is the first geological time period of the Paleozoic Era that lasted from 541 to 485.4 million years ago. During this span of over 55 million years, Earth experienced a dramatic burst of evolutionary changes known as the Cambrian Explosion. The Cambrian is unique for its unusually high proportion of larger strata sedimentary deposits, which are sites of exceptional preservation where both the soft parts of organisms and their more resistant shells are preserved. Consequently, scientists' understanding of Cambrian biology surpasses that of some later periods. The Cambrian marked a profound change in life on Earth. Before this period, most living organisms were small, unicellular, and simple, with the notable exception of Ediacaran fauna. Although complex, multicellular organisms began to appear in the millions of years leading up to the Cambrian, it was during this period that mineralized and thus readily fossilized organisms became common. The rapid diversification of life forms in the Cambrian, known as the Cambrian Explosion, produced the first representatives of most modern animal phyla. Phylogenetic analysis has supported the view that before the Cambrian radiation, in the Cryogenian or Tonian, animals, metazoans, evolved monophyletically from a single common ancestor, flagellated colonial protists similar to modern choanoflagellates. While diverse life forms thrived in the oceans during the Cambrian, the land remained comparatively barren, likely hosting only a microbial soil crust and a few mollusks and arthropods that emerged to graze on the microbial biofilm, although these creatures were not yet fully terrestrial. By the end of the Cambrian, myriapods, arachnids, and hexapods started adapting to the land, along with the first plants. During the Cambrian, almost every metazoan phylum with hard parts, as well as many without, made its first appearance. The only modern phylum with an adequate fossil record to emerge after the Cambrian was the Bryozoa, first known from the early Ordovician. While a few mineralized animal fossils, such as sponge spicules and probable worm tubes, are known from the preceding Ediacaran period, and some Ediacaran biota fossils may represent living phyla, this remains controversial. The Cambrian, however, was undeniably a period of significant evolutionary innovation, with numerous major organism groups appearing within just 40 million years. Trace fossils indicate increased diversity in Cambrian rocks, suggesting that animals were developing new ecological niches and strategies, such as active hunting, deep burrowing, and creating complex branching burrows. Additionally, the Cambrian saw the emergence and diversification of various mineralized algae, including coralline red algae and daisy-clad green algae. Despite the presence of almost all living marine phyla, life in the Cambrian seas would have been quite unfamiliar to a modern scuba diver. Many phyla were represented by classes that have since gone extinct or become less prominent. For instance, while brachiopods were present, the greatest diversity was found among inarticulate brachiopods. Articulate brachiopods, which would later dominate the marine environment of the Paleozoic, were still relatively rare. Cambrian echinoderms were predominantly unfamiliar types, such as early edroasteroids, eocrinoids, and helioplacoids, with more familiar starfish, brittle stars, and sea urchins yet to evolve. The presence of crinoids or sea lilies is debated, but if they existed, they were rare compared to their later Paleozoic abundance. Additionally, while jawless vertebrates were present, it was not until the Ordovician that armored fish became common enough to leave a rich fossil record. The discovery of Precambrian fossils revealed that life evolved long before the Cambrian period. However, the Cambrian is notable for two reasons: the rapid evolution of almost all major groups within about 40 million years, and the rise of animals with hard shells, which greatly improved their chances of fossilization. This explosion of life is especially evident in oceanic fauna, unprecedented in Earth's history. During the Cambrian, land plants had not yet evolved, leaving the terrestrial world devoid of vegetation. In the oceans, marine invertebrates like sponges and brachiopods were present, and the first vertebrates, jawless fish called agnathans, appeared. These heavily armored creatures likely fed by filter feeding as they lacked biting jaws. Today, a few descendants of these jawless fish, such as the lamprey, still survive. One of the best-known Cambrian animals were trilobites, a now extinct group of armored invertebrates. Trilobites thrived in the shallow seas that covered much of the world during the Cambrian, with paleontologists identifying many species. The sediments in which trilobite fossils are found provide insights into their lifestyles, revealing that they included swimming forms, bottom dwellers, and species living in both warm, shallow waters and deeper, cooler regions. Their hard, calcified external skeletons facilitated their preservation in Cambrian sedimentary rocks. Trilobites had flattened, segmented, and plated bodies that provided protection in predator-rich seas. They ranged in size from a millimeter to over 0.6 meters or 2 feet in length, and were among the most successful and enduring prehistoric animals. Over 17,000 species are known to have existed until their extinction at the end of the Permian period, 251 million years ago. In the Cambrian seas, predators like the giant shrimp-like Anomalocaris trapped their prey using fearsome mouthparts lined with hooks. Even stranger was Opabinia, with 5 eyes and a flexible clawed arm attached to its head for catching victims. These creatures hunted along the seabed, where colonies of archaic sponges grew on organic, mineral structures formed by the activity of cyanobacteria. The sponges added to these reef habitats by building supporting skeletons from calcium carbonate, which they collected from the water. Pikaia gracilens, the earliest known primitive chordate, swam in the Middle Cambrian seas. Fossils found in the Burgess Shale of British Columbia show traces of a notochord, a rod-like primitive backbone, a significant step in the evolution of vertebrates. During the Cambrian period, the Earth experienced a generally warmer climate compared to today, characterized by significant fluctuations and regional variations. Evidence suggests the absence of polar ice caps during this time, indicating a relatively warm global climate. However, localized climate patterns were influenced by factors such as the distribution of land masses and ocean currents. Overall, the Cambrian climate was relatively stable, featuring mild temperatures that fostered the flourishing of life, particularly in marine environments. This stability likely played a role in the diversification of marine organisms during the Cambrian explosion. The plant life in the Cambrian was similar to that of the Ediacaran period. The main types were marine microalgae, like Fuxianospira, Cyanocylindra, and Marpolia. There were no calcareous microalgae during this time. No fossils of land plants are found from the Cambrian period. However, there were well-developed biofilms and microbial mats on Cambrian tidal flats and beaches around 500 million years ago. These microbes formed ecosystems similar to the modern soil crust found in desert regions, helping with soil formation. While some estimates suggest that terrestrial plants might have appeared during the Middle or Late Cambrian, significant removal of the greenhouse gas from the atmosphere didn't start until the Ordovician period. High in Canada's Rocky Mountains, the animals of an ancient ecosystem can be seen battling for life. The fossils of the Burgess Shale offer a glimpse at the incredible diversity of early life on Earth, frozen in time and locked in stone. You just have to go digging to see it. Cambrian creatures are all weirdos. For example, Hallucigenia looked like a prickly worm with legs. Yawunik, a half-billion-year-old ancestor of today's insects and spiders, looked nothing like them, and had a head-mounted set of sensory appendages that doubled as claw-like tools for catching and cutting its prey. Among those fascinating and odd-looking critters, there was an absolute monster. Anomalocaris could be considered the T-Rex of the Cambrian. It was the largest predator to actively search out prey to munch on. And as predators go, it certainly looked fearsome. Anomalocaris, unlike other shrimp or abnormal shrimp, is an extinct genus of radiodont, an order of early diverging stem group arthropods. The most well-known species, Anomalocaris canadensis, was discovered in the Stephen Formation, especially in the Burgess Shale of British Columbia, Canada. Another species, Anomalocaris dahliae, was found in the slightly older Emu Bay Shale in Australia, with additional remains found in China and the United States awaiting description. Unlike other radiodonts, Anomalocaris had swimming flaps running along its body, large compound eyes, and a single pair of segmented frontal appendages, which in Anomalocaris were used to grasp prey. Measuring up to 38 centimeters or 1.25 feet long, excluding frontal appendages and tail fan, it is one of the largest animals of the Cambrian, and thought to be one of the earliest examples of an apex predator, though others have been found in older Cambrian Lagerstätten deposits. Since the original description in the late 19th century, the frontal appendages were the only known fossilized parts and misidentified as the body parts of other animals. Anomalocaris moved through the water by undulating the flexible flaps on the sides of its body. Each flap sloped below the one behind it, allowing the lobes on each side to function as a single fin, maximizing swimming efficiency. The design of a remote-controlled model demonstrated this swimming style to be inherently stable, suggesting that Anomalocaris did not require a complex brain to maintain balance while swimming. Anomalocaris' mouth was composed of 32 overlapping plates. Some scientists interpret this as meaning that it could easily crush prey. They point to evidence of its crushing ability, bite-like marks in trilobites. But it has been argued that Anomalocaris' mouth and teeth were too soft to crunch through thick trilobite armor, and the fossil's plates and tips have never shown any signs of wear or tear. This controversy illustrates how hard it is to figure out what animals were doing over 500 million years ago. Based on fossilized eyes from the Emu Bay Shale, the stalked eyes of Anomalocaris were 30 times more powerful than those of trilobites, long thought to have had the most advanced eyes of any contemporary species. With one specimen having over 24,000 lenses in one eye, the resolution of the 3 centimeter wide or 1.2-inch eyes would have been rivaled only by that of the modern dragonfly, which has 28,000 lenses in each eye. Additionally, estimation of ecdysozoan opsins suggests that Anomalocaris may have had dichromatic color vision. The view of Anomalocaris as an active predator has been widely accepted in the history of research, supported by its raptorial frontal appendages and midgut glands, indicating a predatory lifestyle. In the case of Anomalocaris canadensis, its remarkable size among Burgess Shale fauna also establishes it as one of the earliest apex predators known to science. However, the traditional notion that Anomalocaris preyed on hard-bodied animals, particularly its ability to pierce the mineralized exoskeletons of trilobites, has been challenged. Recent studies suggest it was more likely that Anomalocaris exclusively hunted soft-bodied prey. Some Cambrian trilobites show round or W-shaped bite marks matching the mouthparts of Ottoia, previously mistaken for those of Anomalocaris. Further evidence comes from large coprolites containing trilobite parts, attributed to Anomalocaris as the only known organism large enough to produce them. However, Anomalocaris lacked mineralized tissue, making it unlikely to penetrate trilobite exoskeletons. The coprolites might have been produced by other organisms, such as Redlichia trilobites. Another suggested possibility was that Anomalocaris fed by grabbing one end of their prey in its oral cone while using its frontal appendages to quickly rock the other end of the animal back and forth. This produces stress that exploited the weaknesses of arthropod cuticles, causing the prey's exoskeleton to rupture and allow the predator to access its innards. This behavior was originally thought to have provided an evolutionary pressure for trilobites to roll up to avoid being flexed until they snapped. The lack of wear on radiodont mouthparts suggests they did not regularly interact with mineralized trilobite shells, and were possibly better adapted for feeding on smaller, soft-bodied organisms by suction. Anomalocaris canadensis may have been capable of feeding on organisms with hard exoskeletons due to the short, robust spines on its frontal appendages, but this conclusion is based solely on comparisons with the fragile frontal appendages of suspension-feeding radiodonts. Trilobites emerged fully formed upon the Cambrian scene by the time some 521 million years ago that the initial members of this ancient line of arthropods began filling oceanic environments around the globe. They were already creatures with highly developed eyes, complex digestive systems, and admirably functional calcite carapaces. Quite simply, at that moment, trilobites were the most advanced lifeforms that planet Earth had ever produced. Despite their sudden and dramatic appearance in the fossil record, it is obvious that trilobites evolved from earlier, more primitive organisms. Many of these ancestral creatures may have more resembled segmented worms or articulated jellyfish than the distinctive three-lobed animals that we all recognize today. Those possible early ancestors, such as Spriggina floundersi, found in the 550 million-year-old Ediacaran age rocks of Australia, appear to have possessed rudimentary body segments and even primitive genal spines. One of the prime candidates for producing the initial members of the trilobite lineage lies in the remote Siberian outcrops of eastern Russia. There, localized species such as Prophalotaspis and Bigotina appeared to stand as the very base of the entire trilobite family tree. Creatures that more than 500 million years ago provided ample proof that complex, multicellular life could indeed survive on this hostile planet called Earth. The group Trilobita existed from early in the Cambrian period, 520 million years ago, until the end of the Permian period, 250 million years ago. The name Trilobita is derived from the tri-lobed structure of the exoskeleton, which has a raised central lobe or axis and a pair of side lobes, called pleurae. The trilobite body is also divided lengthwise into three regions, or tagmata: a head, or cephalon, a middle region, thorax, composed of several to many articulated segments, and a tail plate called a pygidium, which consists of fused segments. Trilobites unquestioningly belong to the arthropod group, but their exact evolutionary position within the arthropod tree is debated. Early researchers viewed trilobites as primitive anthropods, possibly ancestral to crustaceans and chelicerates, due to their ancient geological history. The presence of a single pair of antennae and a relatively simple leg structure in trilobites suggests primitive traits shared with other arthropods. Recent studies suggest that chelicerates are the closest living relatives to trilobites, evident when comparing them with horseshoe crabs. Trilobites, horseshoe crabs, and sea scorpions share similar spine rows along the inner leg margin. The outer leg branches of trilobites resemble the filaments of horseshoe crab gills and arachnid lungs. Although trilobite eyes penetrate the head shield like those of horseshoe crabs, they are not direct ancestors of horseshoe crabs or other chelicerates. Unique features, such as their calcite exoskeleton and calcified eyes, suggest that trilobites represent a distinct branch within Arthropoda. Some trilobites were blind, probably living too deep in the sea for light to reach them. As such, they became secondarily blind in this branch of trilobite evolution. But many of them had a diverse range of eyes, from simple to highly complex structures with varying lens counts among species. Simple schizochroal eyes had a small number of large lenses, while more complex holochroal eyes had dozens to hundreds of smaller lenses. The most complex, abathochroal eyes had thousands of tiny lenses, with some species boasting tens of thousands. Trilobite eyes could be positioned on stalks or fixed directly to the head shield, showcasing a wide range of adaptations for visual perception in ancient arthropods, with the lens counts varying from just a few to tens of thousands. Trilobites would either hunt or eat worms or other invertebrates, scavenge and eat bits of edible material on the ocean floor, or filter food from the water. Paleontologists interpret what trilobites ate by looking at parts of the exoskeleton that could be used for feeding. Trilobites reproduced sexually, likely having at least 4 different life cycles. One involves laying eggs in a nest on the seabed, where larvae and adults lived on the sea floor, while another method was spawning eggs into the water column, with early swimming larvae transitioning to bottom-dwelling adults. Fossil evidence also suggests eggs were sometimes brooded externally underneath the cephalon, similar to horseshoe crabs, and early trilobite life stages lacking a calcified exoskeleton haven't been found in the fossil record. Like other arthropods, trilobites underwent several developmental stages until maturity. The first mature exoskeleton was small, sometimes as small as the end of a piece of spaghetti. As the body grew, the old exoskeleton was shed and replaced with a new one, cracking along suture lines between parts of the cephalon to allow the soft-bodied animal to escape, resulting in most trilobite fossils being molted exoskeletons, many of which are incomplete. The diversity and geographic range of Trilobita was impacted by 5 mass extinction events, the most significant occurring 375 million years ago, after which only one group of trilobites remained. Trilobites went extinct by 250 million years ago, during the biggest die-off in Earth's history, when 96% of the species on the planet were lost. It is no wonder this event is called the Great Dying. The name says it all. When Charles Walcott first picked out Hallucigenia from the Burgess Shale over a century ago, he was so flummoxed by its appearance that he almost thought he was hallucinating. Heads or tails? Scientists finally determined which end is up for the enigmatic ancient worm Hallucigenia, whose fossils once baffled researchers by appearing topsy-turvy. After decades of study, it's confirmed that Hallucigenia's circular grinning mouth, lined with teeth, is indeed its head. This toothy structure may link creatures as diverse as spiders, nematode worms, and the hardy water bears, also known as tardigrades. Hallucigenia, tiny marine worms from the Cambrian period, measured just 15 millimeters, or 0.59 inches in length. Their spiky fossils, first discovered in the 1970s, revealed a wormy body with spines on top and spindly legs below. However, initial descriptions mistook its spines for legs and vice versa. In a study of Hallucigenia sparsa fossils, researchers found that what was thought to be the head was actually a decay-related remnant, while the true head was at the other end. Using micro-engraving tools, they uncovered dozens of specimen heads, revealing the worm's circular, tooth-lined mouth. This unique mouth structure raises questions about the evolution of feeding strategies in the Ecdysozoa group, to which Hallucigenia belongs. Some members, like tardigrades, have circular, toothed mouths, while others, like spiders, do not. Hallucigenia's tooth-lined mouth suggests it may have fed by suction, but its exact diet remains a mystery. It's speculated that its tentacle-like limbs were used for filter feeding or grasping onto sponges for food. Hallucigenia features a worm-like body with a small head at the end of a long neck and a trunk bearing 7 pairs of long dorsal spines and 7 pairs of slender, leg-like lobes. The spacing between lobes and spines is relatively constant, with the spine pairs shifted forward, leaving the posterior pair of legs without corresponding spines above. Each leg ends in a pair of claws, while the rigid spines have inflexible basal plates. The neck area bears 2 or 3 pairs of very fine anterior appendages lacking terminal claws. Though the head is indistinct, the mouth is anterior, and a straight gut ends in a posterior anus, suggesting the posterior end may be more bulbous than previously thought. Hallucigenia is one of many fascinating creatures found in the Burgess Shale deposits of Canada. The detailed preservation of these fossils offers a window into ancient marine ecosystems and the diversity of life during the Cambrian Explosion. It remains a transitional form between primitive worm-like creatures and more complex organisms. Its body features, such as the arrangement of spines and legs, offers clues about the evolutionary relationship between different animal groups. The creature evolved shortly after the Cambrian Explosion, a period in geologic history characterized by the rapid evolution of various animal groups. The exact period of the Hallucigenia's extinction isn't known, but they probably didn't survive past the Cambrian Ordovician extinction event, which occurred approximately 488 million years ago. Opabinia regalis is an extinct stem group arthropod found in the Middle Cambrian Burgess Shale Lagerstätte 505 million years ago of British Columbia. Opabinia was a soft-bodied animal measuring up to 7 centimeters or 2.8 inches in body length, and its segmented trunk had flaps along the sides and a fan-shaped tail. The head shows unusual features: 5 eyes, a mouth under the head and facing backwards, and a clawed proboscis that probably passed food to the mouth. Opabinia probably lived on the seafloor, using the proboscis to seek out small, soft food. Less than 20 well-preserved specimens have been documented, with only 3 Opabinia specimens discovered in the greater Philipod bed, making up less than 0.1% of the community. Opabinia displayed bilateral symmetry and segmented body parts. Its body consisted of 15 segments, each featuring a pair of lobes facing outwards and downwards. These segments were arranged in a way that each rear end covered the preceding pairs. The conical-shaped tail had 3 overlapping fan blades, forming a V-shaped impression. Respiration occurred through paired dorsal extensions functioning as gills connected to the base of the flaps. Most interpretations suggest that Opabinia, lacking jaw-like structures and teeth, was a carnivore feeding on small, soft-bodied animals, like smaller invertebrates in its habitat. It captured prey with its claw-like proboscis and passed them to its mouth for digestion, utilizing paired gut diverticula to enhance food digestion efficiency. Opabinia regalis was first described by Walcott in 1912 as the most primitive of all Burgess Shale arthropods. Owing to its unique morphology, with a bizarre frontal nozzle, Opabinia became a flagship fossil for the Burgess Shale, leading to much speculation on its affinity and lifestyle. One famous reconstruction shows the animal swimming upside down as an anostracan crustacean. But it looks like Opabinia wasn't just a lonely freak of nature after all. Opabinia regalis is one of the strangest-looking celebrities of the Cambrian period. In fact, this ancient sea dweller is so unique that scientists have never discovered another species in the fossil record that appears to fit into its alien-faced family. But I suggest you meet Eutorora comosa, a small, spiky-tailed marine animal that lived a few million years after Opabinia in what is now North America. First described in 2008, Eutorora comosa was originally classified as a relative of the fearsome Anomalocaris, but a new study suggests that it may have been much more than just another ancient predator. The only known fossil of Eutorora comosa, discovered in Utah's Cambrian Wheeler Formation, had no appendages on its head. Meanwhile, its inch-long body was segmented into 14 or 15 furrows, each tipped with a pointy flap, much like Opabinia. Almost none of its traits fit in with the Radiodont family. Rather, the fossil creature was almost certainly related to Opabinia. This means Opabinia was not the only Opabiniid. New paleontological findings are exciting for a few reasons, not the least of which is that Opabinia can now invite at least one other species to its family reunions. In a broader sense, the existence of another Opabiniid shows that this wasn't just a family of weirdos, but that both creatures were part of a bigger picture of Cambrian evolution. This weird organism, which had a hugely proportioned mouth but no anus, was thought to be a common ancestor of deuterostomes, making it the earliest known ancestor of humans, but it turns out to be on another branch of life. That embarrassing claim about our early ancestor was made back in 2017, but you can relax now, since new evidence suggests the creature, which lived around 530 million years ago during the early Cambrian period, was actually an early ancestor of animals such as crabs and spiders. Fossils of the sack-like animal Saccorhytus coronarius, collected from the Quanshampu Formation in South China, suggest it was around 1.3 millimeters or 0.05 inches long and had a huge mouth, roughly half a millimeter wide, through which it probably both fed and excreted. The organism lived on the ocean floor. Saccorhytus coronarius probably belonged to a major group of animals called the ecdysozoans, which includes insects, crustaceans, and roundworms. The spikes around the mouth helped it to catch prey, although it is unclear what it would have eaten. Living animals like penis worms have the same arrangement of spines, and they use them as part of capturing prey. Basically, the mouth rotates inwards and the spines drag the food material into the mouth. Saccorhytus most likely lived a myofaunal lifestyle, with its body plan suited for an interstitial habitat, such as its thick but flexible cuticle providing protection and allowing it to wriggle through grains of sand. Additionally, it is possible that muscles could have attached to the body cone spines, allowing them to be used for locomotion. Wiwaxia is a genus of soft-bodied animals protected from predators by carbonaceous scales and spines. Fossils of Wiwaxia, which include mainly isolated scales and occasionally complete articulated specimens, have been found in early and middle Cambrian deposits worldwide. The living animal could grow up to 5 centimeters or 2 inches long, with juvenile specimens ranging down to 2 millimeters or 0.08 inches in length. The affinity of Wiwaxia has been a topic of debate among researchers, who were long divided between two possibilities. Some believed its rows of scales resembled those of certain scale worms, annelids, while others pointed to its mouthparts and general morphology as indicative of a relationship to shellless mollusks. Recently, accumulating evidence based on new details of Wiwaxia's mouthparts, scales, and growth history has supported the molluscan affinity. The name Wiwaxia is derived from Wiwaxie Peak in British Columbia. Underneath, Wiwaxia was soft and lacked spines or scales, and little is known about its internal organs. However, there are suggestions it had a feeding apparatus with 2 rows of pointy teeth. The body of Wiwaxia featured 8 rows of sclerites, small armor plates. Most sclerites were oval-shaped, while those closest to the seafloor had a crescent moon shape. These sclerites were not just superficial body coverings, but were buried deep within the organism's body, with up to 40% of their length embedded internally. In addition to these armored plates, Wiwaxia had 2 rows of upward-pointing spines on its body, visible only in larger specimens, suggesting they were absent in juveniles and developed as the organism grew. Despite being unmineralized, both the sclerites and spines were made of a strong organic biopolymer. Experts believe Wiwaxia could regenerate lost spines. The rows of scales and spines likely protected the organism from predators. The closest relative of this animal identified so far is the Odontogryphus omalus. This animal also has a ventral creeping foot with gills around it. Both Wiwaxia and Odontogryphus had molluscan ancestry. There's also a possibility that Wiwaxia belonged to a stem group completely different from any modern family. Wiwaxia was a slow-moving animal and was probably not an active hunter for prey. Rather, it was a bottom feeder. The Cambrian Sea, where this organism lived, didn't have many living organisms to feed on. However, they probably scraped bacteria and other microorganisms off the microbial mat on the prehistoric seafloor. Wiwaxia became extinct during the mid-Cambrian period, likely living between 541 and 485 million years ago. No fossil records indicate its survival into the late Cambrian. While the exact cause of its disappearance is unclear, it is believed that changes in the conditions of the Cambrian Sea, including alterations to the ocean floor, led to the depletion of its main food source, ultimately causing its extinction. A huge discovery, shelved and forgotten for almost 100 years. The remarkable story of the early chordate Pikaia gracilens starts in 1911 when famous Burgess Shale discoverer Charles Walcott finds a strange-looking worm and eel-like fossil in the Canadian Rockies. It's tiny, just 5 centimeters long or 1.9 inches, and nowhere near as exciting as the massive Cambrian arthropod monsters he'd found thus far. So Walcott marks the fossil as a worm and shelves it. And it takes mankind until the 1970s to find it, re-examine it, and realize that Walcott's worm was no worm at all. It's the then first known Cambrian chordate, the ancient precursor to vertebrates, all creatures with a backbone, from Dunkleosteus to Velociraptor, to ground sloths, and all the way to mammals and us humans. The most distinctive feature of Pikaia is its notochord, a flexible rod-like structure that runs along its back, which is a key characteristic of chordates. This notochord is thought to have been used for swimming, supporting the body, and as a precursor to the vertebral column in later vertebrates. We only have evidence of Pikaia in one slice of time, the Middle Cambrian period, specifically around 505 million years ago, a time when life in Earth's oceans was becoming more diverse and complex. We haven't found any other evidence, fossils of Pikaia, in any previous or subsequent rock layers, so we have to assume it lived and went extinct in the Cambrian. Scientists think Pikaia must have evolved from simpler, non-chordate ancestors. These precursors were likely similar to other early multicellular organisms found in earlier strata, such as Ediacaran, and they probably lacked the defining characteristics of chordates, such as a notochord and a dorsal nerve cord. While we don't know the exact diet of Pikaia, scientists speculate that it was a filter feeder or scavenger, consuming small particles of food or organic matter suspended in the water. You can get the idea of its potential feeding habits by looking at its closest living modern relative, the cephalochordates, lancelets, who are filter feeders. The fossil evidence of Pikaia does not clearly show any eyes, leading scientists to speculate that it might have had simple light-sensitive spots instead of well-developed eyes. This would have been sufficient for detecting changes in light intensity, helping it navigate the Cambrian seas. Is Pikaia considered a fish? No, Pikaia is not classified as a fish. While it shares some characteristics with modern fish, such as a notochord and a basic body plan, it lacks other defining features of true fish, such as jaws, fins, and a complex skeletal structure. The extinction of Pikaia, like many species from the Cambrian period, is attributed to natural selection and environmental changes that made survival challenging. Over millions of years, evolutionary pressures and changing ecosystems likely favored the emergence of more advanced life forms, outcompeting Pikaia and leading to its eventual disappearance from the fossil record. Marrella is an extinct genus of marrellomorph arthropod found in the Middle Cambrian deposits of North America and Asia. It is the most abundant animal species in the Burgess Shale of British Columbia, Canada, with tens of thousands of specimens collected. Although much rarer, remains of Marrella are also known from deposits in China. Marrella was the first fossil collected by Charles Walcott from the Burgess Shale in 1909. Initially, Walcott informally referred to Marrella as a lace crab, and later described it more formally as an unusual trilobite. However, it was later reclassified to the now defunct class Trilobitoidae. In 1971, Whittington conducted a detailed re-evaluation of the animal and concluded, based on its legs, gills, and head appendages, that it belonged to neither trilobites, chelicerates, nor crustaceans. Marrella is among several unique arthropod-like organisms found in the Burgess Shale, alongside Opabinia and Aysheaia. The diverse and peculiar characteristics of these creatures were quite surprising at the time of their discovery. Their fossils provided evidence that the soft-bodied Burgess fauna was more complex and diverse than previously thought. Specimens of Marrella vary in size from 2.4 to 24.5 millimeters, or 0.09 to 1 inches in length. The head shield featured two pairs of long, backward curving projections or spines, with the posterior pair having a serrated keel. No evidence of eyes has been found. Beneath the head, there were a pair of long, flexible antennae composed of approximately 30 segments, projecting forward at an angle of 15 to 30 degrees away from the midline. Adjacent to and slightly above the antennae, there were short, stout, paddle-like swimming appendages attached, consisting of one long basal segment and 5 shorter segments, the edges of which were fringed with setae. Marrella is likely to have been an active swimmer that swam close to the sea floor, nectobenthic, with its swimming appendages used in a backstroke motion, with the large spines acting as stabilizers, as well as possibly having a defensive function. They have been suggested to be filter feeders, with food particles sifted out of the water column by the posterior appendages during swimming before being passed forward by the appendages towards the mouth. Marrella is one of the most common species in the Burgess Shale. Over 25,000 specimens have been collected, and it is the second most common arthropod species in Walcott Quarry, comprising 7.3% of the specimens counted. Bigger and slightly easier to study, Metaspriggina walcotti is a basal or stem vertebrate of about 10 centimeters or 3.9 inches in size from Canada's famous Cambrian Burgess Shale that shows a clear transition from the basic chordate body plan to that of early vertebrates. Unlike crown group vertebrates, Metaspriggina displays pivotal body features that show it's a transitionary form between chordates and vertebrates, underlining its significance in the evolutionary narrative. These features include the chordate notochord, primitive spine, dorsal nerve cord, and postanal tail, along with the beginnings of vertebral elements, possible lobe-like extensions in the head, primitive brain, fish-like myomeres, muscle segments, a primitive brain case, and upward-facing eyes, presenting a clear depiction of its transitional form between earlier chordates and true vertebrates. Metaspriggina's anatomy is strikingly fish-like, setting it apart from other stem vertebrates of its era in both appearance and size. Its body was streamlined, suggesting an adaptation to a more active lifestyle in the Cambrian seas. The presence of W-shaped muscle bands along its body suggests this fish was a strong swimmer, although it may not have had preserved fins. Its feeding behavior and eye position suggest it lived near the sea bottom, where it likely detected predators. This is supported by the abundance of fossilized Metaspriggina specimens found in former seabed mudflows. Ottoia is an early archaeopriapulid worm found in Cambrian fossils. While priapulid-like worms from different Cambrian sites are sometimes labeled as Ottoia, clear Ottoia macrofossils are mainly from the Burgess Shale of British Columbia, dating back 508 million years. Microfossils also extend Ottoia's record across the western Canada sedimentary basin from the mid to late Cambrian. Some fossil finds are also documented in China. Ottoia specimens typically reach 8 centimeters (3.1 inches) in length, with variations in width due to contraction. Shorter individuals are often wider than longer ones. The characteristic proboscis of priapulids is located at the anterior end, attached to the trunk, followed by the bursa at the posterior. While the body is bilaterally symmetrical, the anterior displays external radial symmetry. Like some modern invertebrates, Ottoia is protected by a cuticle that also limits its size. Ottoia was a burrowing predator that captured prey with its eversible proboscis. It's also believed to have scavenged dead organisms like the arthropod Aysheaia. The spines of Ottoia's proboscis were likely used as teeth to catch prey. While its exact lifestyle is uncertain, Ottoia is thought to have actively burrowed through sediment in search of prey, likely living within a U-shaped burrow it constructed. From this sheltered position, it extended its proboscis to hunt. Gut contents reveal it primarily preyed on hyolithid Haplophrenites, swallowing them head first, and showed signs of cannibalism, a behavior common in priapulids. Canadaspis is a remarkable genus of early arthropod that flourished during the Cambrian period, over 500 million years ago. Its fossils have been found primarily in the Burgess Shale of British Columbia, Canada, offering scientists invaluable insights into the early evolution of arthropods and the diversity of life during the Cambrian Explosion. Canadaspis fossils were first discovered in the early 20th century by Charles Walcott, alongside other iconic Burgess Shale organisms. The genus name, Canadaspis, translates to Shield of Canada, reflecting the distinctive appearance of its exoskeleton. Canadaspis specimens vary in size, with some individuals measuring only a few centimeters in length, while others reach up to 10 centimeters or 3.9 inches. Its body was covered by a bivalved carapace, resembling a protective shield, giving it a distinct appearance among other Cambrian organisms. Its unique bivalved carapace likely provided protection against predators and environmental hazards. This carapace was composed of 2 separate shells, hinged along the dorsal midline, allowing the organism to open and close its armor for feeding or defense. Beneath its armored exterior, Canadaspis had a segmented body, typical of arthropods, with jointed appendages. However, unlike many other arthropods, Canadaspis lacked specialized appendages for swimming or walking. Instead, it likely moved along the seafloor, using its appendages for feeding and possibly burrowing. Its flattened body and broad carapace suggest an adaptation to life in shallow marine environments, possibly in close proximity to reefs or other structures. Feeding strategies of Canadaspis remain a topic of debate among scientists. Some propose that it was a suspension feeder, using its appendages to capture plankton or detritus from the water column. Others suggest it may have been a deposit feeder, using its appendages to sift through sediment for organic matter. Canadaspis provides insights into the ecological roles played by early arthropods during the Cambrian period. Its presence in the Burgess Shale suggests it was part of a complex ecosystem, interacting with various other organisms in the ancient seas. Orthrozanclus is a genus of sea creatures known from 2 species. Animals in this genus were 1 or 2 centimeters long, with spikes protruding from their armored bodies. The placement of this genus into a specific family is not universally accepted. The genus name means straight rod or upright wall, referring to its unique morphology. Orthrozanclus is characterized by its flattened, oval-shaped body, covered in rows of armor plates or sclerites. These sclerites are arranged in a distinctive pattern, forming a series of parallel rows along the dorsal side of the organism. At the anterior end, Orthrozanclus features a pair of elongated tentacles or appendages, thought to have been used for feeding. Beneath its armored exterior, Orthrozanclus had a soft body with a simple gut and musculature. Its appendages likely played a role in feeding, possibly capturing small organisms or detritus from the water column. The ecological role of Orthrozanclus remains a subject of debate among scientists. Some propose that it was a filter feeder, using its tentacles to capture plankton or organic particles from the water. Others suggest it may have been a scavenger, feeding on detritus or carrion on the seafloor. Orthrozanclus is believed to have inhabited shallow marine environments, possibly living near the seafloor or attached to hard substrates such as rocks or coral reefs. Its flattened body and armor plates likely provided protection against predation and may have helped stabilize the organism in turbulent waters. Studying Orthrozanclus and other Burgess Shale organisms continues to deepen our understanding of ancient ecosystems and the origins of modern animal groups, highlighting the rich tapestry of life's history on our planet. Isoxys is an intriguing genus of extinct arthropod that thrived during the Cambrian period, approximately 520 million years ago. Fossils of Isoxys have been discovered in various locations around the world, shedding light on the early evolution of arthropods and the diversity of life during this ancient time. The first fossils of Isoxys were discovered in the early 20th century, primarily in Burgess Shale deposits in British Columbia, Canada, but specimens have also been found in other Cambrian sites globally. The name Isoxys means equal foot, reflecting the symmetrical arrangement of its legs. Isoxys was a small organism, typically ranging from a few millimeters to several centimeters in length. Its body was elongated and segmented, resembling a primitive form of arthropod with a cylindrical shape. Each body segment bore a pair of jointed appendages, suggesting a close affinity to early arthropods. Its segmented body was covered in a tough exoskeleton, providing protection and support. The head of Isoxys featured a pair of antennae or sensory organs, indicating its ability to detect changes in its environment. Despite its small size, Isoxys likely had well-developed sensory capabilities, allowing it to navigate its surroundings and locate food sources. A predatory lifestyle is indicated by the large eyes, frontal appendage, and gut glands. Isoxys would have swum just above the seafloor, seeking out prey in the water column and at the sediment-water interface. Nectocaris is a perplexing genus of squid-like animal with uncertain evolutionary relationships, known from the Cambrian period approximately 500 million years ago. Initially discovered in the Burgess Shale of Canada, Nectocaris fossils have sparked debates among paleontologists due to its unique characteristics. The first fossils attributed to Nectocaris were unearthed from the renowned Burgess Shale deposits in British Columbia, Canada. These fossils reveal an organism with a streamlined, elongated body, resembling a primitive cephalopod or squid. However, its exact classification has been a subject of controversy. Similar remains, potentially belonging to the genus Nectocaris, have been found in other Cambrian deposits, including the Emu Bay Shale of Australia and the Chenxiang biota of China. These additional discoveries suggest a wider distribution of Nectocaris-like organisms during the Cambrian period. Nectocaris exhibits several features typical of cephalopods, such as a streamlined body, tentacle-like structures, and possibly jet propulsion for locomotion. However, it also possesses characteristics that challenge its classification within this group, leading to ongoing debate among scientists. The most intriguing aspect of Nectocaris is its eyes, which are among the oldest complex eyes known in the fossil record. These eyes were composed of numerous individual lenses, suggesting a high degree of visual acuity for an organism of its time. Aysheaia is a fascinating genus of extinct worm-like creature that inhabited the Earth during the Cambrian period, approximately 520 million years ago. Aysheia's body was elongated and segmented, with each segment bearing a pair of stout, pointed spines. Its head was equipped with a pair of antennae-like structures, possibly used for sensory perception or feeding. It had a soft body covered by a tough outer cuticle, providing protection and support. Its segmented body allowed for flexibility and movement, while the stout spines likely served as defense against predators or environmental hazards. Recent studies on Aysheaia have revealed intriguing details about its reproductive biology. Fossil specimens of Aysheaia have been found in association with clusters of small eggs, suggesting that it may have reproduced by laying eggs in protected environments. Additionally, some Aysheaia fossils show evidence of preserved gut contents, providing insights into its diet and feeding habits. Analysis of these gut contents indicates that Aysheaia likely fed on organic matter and small organisms that lived within the sediment. Amiskwia is a genus of extinct marine organism known from fossils dating back to the Cambrian period, approximately 505 million years ago. Due to its relatively simple appearance, Amiskwia holds a special place in paleontological studies due to its unique morphology and uncertain evolutionary relationships. Amiskwia's body was segmented with numerous repeated units along its length. Its most striking feature is the proboscis, which extends from the front end of the body and may have been used for feeding or sensory purposes. Its segmented body allowed for flexibility and movement, while the proboscis likely played a crucial role in feeding or detecting prey. One of the most intriguing aspects of Amiskwia is its tentacle-like structures, which extend from the sides of its body. These structures may have been used for filter feeding or capturing small organisms from the surrounding water. Some Amiskwia fossils show evidence of preserved gut contents, providing insights into its diet and feeding habits. Analyses of these gut contents indicates that Amiskwia likely fed on organic matter and small organisms that lived within the sediment. The advancement of the theory of glaciation as the predetermining agent for the Cambrian extinctions has been developed by James Miller of Southwest Missouri State University. Through research undertaken by Miller, evidence of early Ordovician sediment of glacial origin has been uncovered in South America. Miller suggests in his hypothesis that this evidence of continental glaciation at the Cambrian-Ordovician boundary is responsible for a decrease in global climatic conditions. Such a decline in temperature is implied by Miller to destroy Cambrian fauna, which are intolerant of cooler conditions, producing a mass extinction of mostly warm water species. He also suggests that a significant continental glaciation would bring large amounts of ocean water onto the land in the form of frozen glacial ice. This trapping of ocean water inevitably results in the decrease of sea level and the withdrawal of shallow seas. Miller implicates that this reduction in sea level would produce reduced habitat for marine species as continental shelves are obliterated. Ecological competition would consequently ensue, perhaps acting as a driving agent for extinction. The development of a hypothesis invoking the cooling and depletion of water in marine waters as a causative agent for the Cambrian extinctions has been advanced by several geologists, primarily Alison Palmer and Michael Taylor of the U.S. Geological Survey and James Stilt of the University of Missouri. The cooling and oxygen depletion would occur when cool waters from deep zones of the ocean spread up onto the continent, eliminating all organisms not able to tolerate cool conditions. The cooling would also result in stratification of the water column. Thus, species would ultimately perish due to their inability to tolerate dramatic shifts in such limiting factors as temperature and oxygen availability. Further research is required to more fully test the validity of the above outlined Cambrian extinction hypotheses.